Day 1.
We arrived bright and early. After a while of waiting, we got to see the ground floor and then made our way up the double helix staircase. We went into the biodiscovery lab where we learned how to use micropipettes. There was a quick pipette challenge where you had to figure out how much colored water was in the mystery epi. We then went on a tour around level 1 labs and level 7. The food was great and was well catered. We then found out about the HeLa story and discovered about the bioethics around it. We then did cell staining and learned about other career paths in medical research.
Day 2. We started the day off learning about PCR. We had a quick game trying to replicate this process and it got pretty hard by round 3. We then went into the lab to try to prep for our gel electrophoresis. When we were loading the wells, it was very stressful, but luckily none of ours were contaminated. We then went through substitution mutation and how we can figure out the wrong nucleotide through sequencing.
We also had a speed date with 3 uni students, Alexis, Angel and Ellie, where we had the opportunity to ask questions about their high school life and uni life, as well as what their science degree encountered. This offered us insight to what life was like beyond high school. Day 3. On the 3rd day, we went straight into the lab and started off by learning about fluorescence. This required the use of UV light to observe a sample of colon cancer cells. We then moved on to an interesting lab about disease outbreak. This simulation showed us how, theoretically, a disease could spread throughout a wider community. It involved sharing a solution throughout the group with 2 other people that acted as blood. One person was unknowingly infected and spread Ebola to the rest of the group. We then tested our EPIs using ELISA to determine if IgM was present in our EPIs. If our EPIs turned purple, then we were infected.
After this, we had a quick stop for lunch and then moved on to testing proteins using seafood. 2 hours flew by, and after gel loading, we were able to compare the proteins for the unknown sample to the seafood.
Today we spent the day in the lab learning about restriction enzymes that help segment DNA. Over the course of 4 days we learnt so much, met new people, played among us, ate delicious food and had fun. Our experience at Harry Perkins was unforgettable.
Thank you to the Fogarty Foundation for giving us this opportunity.